if we're going to have a chance of conserving plant biodiversity in a changing world, and if we're going to have a chance of using plant diversity to feed that world, then to my mind we need to understand the diversity that's there. We need to understand how it's built, and we need to understand what it's for. And my lab is interested in questions of understanding how come there are so many different sorts of flowering plants, and how are they built, and what are they there for? Now, this isn't a question that's new. Darwin noticed it and wrote in this famous quote to Hooker that the rapid development of the angiosperms was an abominable mystery. And what he means by that is that the angiosperms appear very rapidly in the geological record, and there are many, many species of them, and far more species of them than of any other group of plants. Now, if you're interested in thinking about angiosperm diversity, you can think about this from a whole range of different angles. The angle we take in the lab is to think about flowers and the interaction between those flowers and the animals that pollinate them. And there are two real good reasons for this. The first is if you just look at angiosperms, it's the diversity of flowers you see. So a lot of the morphological variation in the reproductive structures of flowering plants is what taxonomy, classification are based on. We see that morphological diversity as really quite extreme. But the other good reason for it is that if you're going to divide a population of organisms into multiple species, you need to break them up into reproductively isolated groups. It's very hard for plants to achieve reproductive isolation unless they recruit an animal which has behavior and preferences to carry their pollen around for them. And so by recruiting animals as pollen vectors, the angiosperms, probably accidentally, ended up with many reproductively isolated subpopulations and therefore many different species. So we're interested in this link, how the variety of floral forms is produced and how they affect interactions with pollinators. Now, aside from the reasons we're interested in that, there are also some good practical reasons. So here are a range of crop plants that are all dependent on animal pollination for their yield. So we think, of course, of the cereals as being wind pollinated, but in fact all of most of our fruit and nut crops, and indeed many of our oilseed crops too, are heavily dependent on animals for pollination. And these are a range of crops that in different parts of the world at the moment are experiencing limited crop yield believed to be the result of variation in pollinator availability at different times and places. So if we can understand that link, then we again have perhaps an opportunity to use that understanding to maximize pollinator attraction to the key crops that we need. Now, we focus on this question of angiosperm diversity from many different angles, and we bring them together, I think, in a diagram that looks a bit like this. So my background is in molecular genetics of development, and we try to find floral traits that we can dissect the developmental basis of, even though they're not perhaps in conventional plant molecular genetic models. The tools that are available today, particularly through transcriptome and genome analysis, mean that there's much more ability to access different species with different and diverse morphologies than perhaps there has been in recent years. But we're also interested in the function of these traits, and so we keep a second lab where we use the tools of behavioral ecology with caged pollinators to try and understand how these traits function and why they're maintained across evolutionary time. Now, if you understand how something's built and why something's built, you can put those two things together and understand how it changes. And so you might perhaps map the trait onto a phylogeny here and see the trait changing at this point and this point, and ask yourself questions about the molecular basis of that change and the functional basis of it, and whether it's related to pollinator shifts or changes in pollinator behavior. So I'm going to show you some of our work using this triangular approach today, focusing on the petal epidermis and on two particular aspects of that petal epidermis. So here's a scanning electron micrograph. This is a Speedwell petal epidermis, but it's fairly representative of many angiosperms. So you see these cells are cone-shaped or papillate. They're protruding out of the board towards you. That's a very common petal epidermal morphology. It's used by developmentalists as a marker for petal identity, as it's not found generally in many other parts of the plant. But the other trait I want to talk about are these patterns on top of the cells, these folds of the cuticle, which you can see are producing stellate patterns on each cell, but actually run across cell junctions in various places on my image. And I'm going to talk about both of these traits and how they interact to attract pollinators to the flower. So I'm going to start with the cuticle patterning, and this is more recent work on which we have less data. I won't be focused so much on the evolution in this, just on understanding the development and the function. So the species we've been working with as our model for this is Hibiscus trionum. It's not perhaps a conventional molecular genetic model, but actually it's very useful. Its seed to seed time is about 6 to 8 weeks, which might be familiar to the Arabidopsis people, and it is very easy to grow in the lab, although obviously much bigger than Arabidopsis. Now it has white and red petals. You see the white on the outer part, red in the middle. But if you look closely at the red, and it's hard to see in this light, the red is anthocyanin pigmentation, but overlying that you can just about make out yellows and blues and greens. It looks like petrol on a puddle. If you look at the junction between the white and red regions of the surface, the white region here has smooth epidermal cells, the red region has these folds of cuticle on it, and it's the folds of cuticle that are generating not the red, that's pigment, but the petrolly oily appearance on top. Now this oily appearance we call iridescence. If you move the flower and change the angle at which you view it, it looks different colours. And at this stage people are often thinking, well, I haven't noticed flowers do iridescence very much, so I just want to show you a couple of other examples of familiar iridescent species. This is a tulip variety called Queen of the Night. Again, you can see the yellow, green, blue, very different colours from different angles. But it's not always immediately obvious if the pigment swamps it. So this is a South African species called Grelum. It looks very yellow and metallic, but if you peel the epidermis off and put it on a microscope slide and change the angle, as you change the angle you see different colours of light reflected. And that's because of the iridescence coming off that epidermal surface. Now we can measure that iridescence. It's fairly straightforward to measure. These are conventional spec readings of light reflection from the petal epidermis, looking at wavelength along the bottom, percentage reflectivity up the side, and you would, for instance, say that the purple line is a spec reading of a flower that's blue. Its peak reflectivity is in the blue wavelengths, and the yellow line is a spec reading of a flower that's yellow. Its peak reflectivity is in the yellow region, but actually these are all readings of the same tulip. All we've done is vary the angle at which we're collecting the light that comes off that cast. And so we're able to measure the iridescence and record which regions of the spectrum it's strongest in. Now, this iridescence is produced by the folds of the cuticle, and I need to just show you how they work. They work in the same way that a CD works. So if you look at a CD, it's iridescent. If you move it around, you see different colours of light. If you look at it on the SEM, you see these data grooves here. And the data grooves happen to be spaced at just the right frequency to interfere with light when it arrives at the surface and to cause constructive interference of different wavelengths at different angles. So blue light's constructively interfered that way, and you see blue at that angle. Red light's interfered with that way, and you see red at that angle, and so on. So what you see here is that what you get if you shine white light on a transparent epoxy replica of the surface of a CD. So colour is produced from an entirely transparent material because of the way this structure interferes with the light. Now if you look at the surface of the tulip on the same scale, you find that those folds of cuticle are again on the same scale as those of the CD, and they're interfering with white light in the same way. They're not quite as good. The tulip is curved, and of course if you look at it in cross-section it's not as perfectly regular, but it's pretty good at producing colour from a transparent material. Now the ability to produce colour from a transparent material is called structural colour, and it's something that's well known in the animal kingdom. It's been studied in butterflies, fish, birds, spiders, various other insects as well. The blue morpho butterfly is perhaps the most famous example. This butterfly has no blue pigment at all. It's simply reflecting blue light through a surface structure, giving you this bright blue impression. But if you look at the literature on structural colour in animals, it's focused on two things. It's focused on understanding the physics, so what the structures are and how they interfere with the light. And it's focused on understanding what those structures do, so how they function as signals in species recognition, in mate choice. There's very little work on understanding how those organisms build structures that are regular enough to interfere with light in a reliable enough way that they can use them to choose mates. That's what we're trying to work out with the Hibiscus system. So we're trying to understand how the living material produces this structure, which is known as a diffraction grating, the structure that can generate the iridescence. Now, our diffraction grating is produced from the cuticle. And just to remind you, if you haven't thought about the plant cuticle for a while, it's the cutin monomers are produced within the cell. They're secreted across the cell membrane and cell wall and assembled out here on the surface of the cell into a series of layers with various amounts of wax, cutin, pectin. And there are usually waxes out on the surface as well. Now, our diffraction grating isn't composed of these waxes. It's composed of folds of this whole structure. So you have to imagine this in waves. And so to understand how that's built, we could think about complicated issues about how the cutin monomers are secreted in the right place, how the thing is assembled in the right way. But actually, it's easier to think about it from a simple physical process thinking about how much cuticle is produced and how the timing of that cuticle production relates to the timing and directionality of cell growth. And I want to just show you a model that we produced out of this, and this is some work that we produced last year in relationship to the Nottingham Maths for the Plant Sciences Study Group, which I recommend if you want to get some mathematicians involved in your work. So if you think about the cell producing cuticle, the cuticle is a amorphous, viscous mass that's secreted out through the cell, and it's not itself living. Now, if the cell grows anisotropically, more in one direction than another, the cuticle will continue to be produced, but spread. I think of it as a bit like treacle on top of a treacle sponge pudding. So you get this spreading viscous layer. Now, that creates friction forces because the boundary between the cell, the top of the cell, and the underneath of the cuticle is under pressure, and you get compressive forces inwards and stretching forces outwards. And if you take a piece of cling film and stretch it in that way and compress it in the other way, what you'll find you get are folds in it that very much mirror the folds that we find in the cuticle of these iridescent flowers. And in fact, just by playing around with the timing of cell growth and the amount of cuticle production, we're able, at least in theory, in the model, to generate all of the patterns of cuticular patterning that we see in the different species of angiosperms. Now, coming back to our hibiscus, it had actually, if you remember, an iridescent region and a non-iridescent region. And if we look at those, I hope you can see this, there are 3 stages, stage 1, 2, and 3, where we can't tell those cells apart. If we look at them on the confocal microscope, the cell shape is the same. But at stage 4 and at stage 5, the cells are clearly different because the cells that are going to become iridescent grow primarily in what you might think of as the y-axis, this direction, whereas the cells that are not grow in the z-axis, so towards you. They're growing out of the board as conical cells. You see that more clearly down at stage 5. So we have differences in growth of the cells matching whether they're going to be iridescent or not. And if we look at the timing of that, then in fact the timing at which we can tell the cells apart on the confocal microscope was this stage 4, and that's the first stage at which we see iridescence, in which we see the diffraction grating produced. So at least in theory at the moment, all of our data matches this model and suggests that by simply understanding how the amount of cuticle is controlled and its timing relative to the direction of cell growth is controlled, we should be able to understand how the plant makes this diffraction grating. Now, I'm going to come back to cell shape in a bit. I just want to briefly talk about cuticle. We have some suggestions of how cuticle production is controlled, and this is work from Asaf Aharoni's lab in the Weizmann Institute in Israel. So he showed that a group of transcription factors in Arabidopsis called the SHINE genes, and SHINE is a subgroup of the AP2 transcription factor family, are responsible for regulating the transcription of the genes encoding the enzymes of cutin monomer synthesis. So you essentially need SHINE to turn on cuticle production. And if you overexpress it in Arabidopsis and you get too much cuticle, that cuticle often folds in ways that mimic the patterns we see on our iridescent flowers. Now, I started this talk by saying that it's possible to work in some very non-model species these days. So Hibiscus trionum doesn't have a genome sequenced. We can do transcriptome work and look for SHINE genes. But in fact, before we do that even, we need to know how many we're looking for, because a transcriptome is not necessarily comprehensive. And so we produce these phylogenetic trees of all of the sequenced SHINE genes from any published database. And by doing that, we're able to establish that there are 2 duplications in this gene family, producing 3 eudicot clades. And so we know we should be looking for 3 genes in our hibiscus plant, whether we do that through transcriptome or through your conventional degenerate PCR. In fact, hibiscus only has 2, and we're comfortable with that because so does cotton, which is in the same family and has a sequenced genome, so cotton has lost the SHINE C clade member, so has our hibiscus. It's probably a family-specific loss of that duplicate. And having identified the genes, we're then able to do conventional molecular genetics. We can use qPCR to assess timing of transcription of these genes. We can transform this species. It's readily transformable, so we can knock them out. We can play around with their timing and amount of expression relative to cell growth, and are able then to assess how these things work together to generate the structures I've shown you. Now, that's an understanding of how you make iridescence, but also I want to understand, I said, what it's for. And we do that using this sort of second lab I have, a bee behavioral lab. And this is a very low-tech setup. So we buy colonies of Bombus terrestris, buff-tailed bumblebee, and they're sold for tomato pollination in greenhouses. We connect them with a gateable tube to to a plywood box with a perspex lid. Now when they come to us, they've never met a flower, and so we persuade them that this is the world. Just like in the real world, they have to leave the nest, they have to fly out here, they forage, they find food, they take it back home. And while they're doing that, we mark them with paint. So we can use different colour paints. You see blue here or red here to mark individual foragers, and that gives us very useful, statistically malleable data. Now, this is the sort of artificial flower we might use in a training bout. We've got an upside-down Eppendorf cap here with sucrose solution in it. The bees think that's a flower, they come out, they feed, they go back to the hive. And we can use this setup to ask various questions about what the animal can perceive, what it might prefer, how it handles different things, and how those issues come together to affect its foraging preferences and behaviour. So for instance here, I'll very briefly show you some data that tells us that bees can see iridescence, and they can learn to use it as a cue to identify rewarding flowers. So in this experiment, the bees have been asked to work out that iridescent blue and yellow and purple discs have sucrose solution, have a reward, and non-iridescent purple and blue and yellow discs don't. And they've been asked to learn that trick. Now the first 10 visits or so they make to these flowers, they get it right about half the time. They haven't worked it out yet, but over time they become 80% accurate at identifying rewarding flowers just on the basis of the iridescence. So we know they can use it as a cue, they can learn it, they can see it, and in fact it increases the efficiency of their foraging. So this is time to find a second flower. This is a different experimental setup. Once you've found a first, and if you add a diffraction grating to a red flower or to a blue flower, you increase foraging efficiency and reduce the time it takes the pollinator to find that flower. So by using these kinds of approaches in combination, we're able to get at questions of how a trait develops and why a trait develops. Now I said at the beginning, I'd like to bring those together and think about how traits change over time. And that's not something we've yet started doing with the iridescence. We're still in the early days of that work. But we've had a lot more time to work on the base shape of the cells of the petal epidermis, and so I'm going to skim through the development and function of these and just tell you a little bit about their evolution. So the development is very old work. This comes from my PhD, actually. And essentially, using mutagenized lines of antirrhinum, we were able to identify a gene called Mixingture, which is responsible for the production of those cone-shaped cells. Here's an unstable mutant where a transposon is excising somatically. So here's the mutant tissue, here's the conical-celled wild type. And so that Mixingture gene encodes a MIB transcription factor. And although in antirrhinum it's only expressed in the petal epidermis, if we express it ectopically in other tissues, it's able to convert, say here, tobacco leaf epidermal cells from flat through to cone-shaped. So it's a master regulator of epidermal cell outgrowth. Now the function of those conical cells on the petal is to do with pollinator grip. So if you look at the bee in close-up, you can see that it has tarsal claws down here. And these claws interlock in between the conical cells. They're on the same scale, and the presence of conical cells on a petal increases the friction and improves the grip the pollinator has on the surface. So we find that they prefer that increased grip, again, because it increases the energetic efficiency of foraging. So if we make an epoxy replica surface, so this is an SEM of this epoxy disc, which is a flat-celled epoxy disc. So it's a replica of a flat-cell petal. And we make equivalent conical-celled ones, and we offer them to the bees held horizontally. There's a colour code on this, and it doesn't really matter. So either you have conical-celled ones purple or conical-celled ones pink. It doesn't matter. Two different experiments. We find there's no real preference. The bees don't care what shape the epidermal cells have. But if we tip those discs up, so the bee has to land on a vertical surface and get a grip on it and forage, then we get a much increased preference for the conical cell form. And if we look at that in slow motion, it's because the bees are unable to grip these flat-celled discs properly. The wings have to keep beating to maintain balance, and so energetically, foraging isn't as beneficial on a flat-celled disc as it would be on a conical-celled one. Now, this is quite a common form. I told you at the beginning that developmentalists use the conical cell form as a marker for petal identity in flowering plants. So we were interested to know when it first evolved and how ancient it is within the angiosperms. So we had a look down in these early diverging clades of angiosperms, and we find that, well, Amborella does have conical cells, but they're a little bit strange. I want to skip past that and tell you about Kabomba here, which is in the Nymphales. So this is an early diverging water lily, and Kabomba has very beautiful conical cells and makes a reasonable model for us to work with. And so we've been able to identify the Mixture ortholog in Kabomba and find that if we express that in tobacco, we're able to convert flat cells through into cones and in fact full multicellular trichomes, just like the U-dicot Mixture gene I showed you earlier could. So it seems that this trait is ancient and the genetic control of this trait is ancient, which would lead you to think that all plants would have it. But in fact, around 20% of petaloid angiosperms don't have conical cells on those petals. And that seems strange to me. If it's an ancient trait, it's a useful one. Why doesn't everybody have it? So we've been doing some work focusing on the genus Solanum, trying to understand there how conical cells have come to be lost. So here's Solanum dulcamara, woody nightshade, bittersweet. It's a common European weed, and it has flat petal epidermal cells. Now, like all the other members of its genus, it's pollinated not by bees foraging for nectar, which it doesn't really produce, but by bees foraging for pollen. So these enlarged yellow anthers dehisce through pores at their tips. Pollen is released through the pores, the bee grabs the anthers and vibrates them, and the pollen flies out onto its body and it takes it back to the hive to feed to the larvae. So you might argue that there's a selective advantage in having slippery petals. Maybe you don't want grip on the petals because the focus of the pollination system is here. Or you might argue that perhaps it's just genetic drift. It makes no difference whatsoever to this flower whether its petals are grippy, or good to grip or hard to grip, because the bee isn't interested, it's gripping onto the anthers. I'll come back to that in a minute. Now, if you look across the genus Solanum, you find dulcamara is only one example of many. So it's a very big genus, there are 1,500 or so species, and each of these arrows is an independent loss of the conical cell form. If I focus in on the Leptostomonum clade up the top there, show you how we do that. So each of the species with a black box next to it has flat petal epidermal cells. The white boxes are conical ones. By mapping the character onto the phylogeny, you're able to get a good idea of how many times the trait shifts. So independent losses here, 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 and probably about here. So it's an event that's occurring frequently at the molecular level. It's not a single loss that's been preserved. It's lost many times over within the genus. Now, to try and understand how that's happened, we need to go back to the molecular work. And so if we isolate the Mixture-like genes from Solanum dulcamara, we find that they are expressed not in the petals at all, but in the leaves, in contrast to a conical-celled relative, there's Petunia. And this is true of all of the flat-celled species we've studied. So here are two others in purple and green. The blue is a conical-celled species for comparison, and this is qPCR data of mixture gene expression. And if you look in the floral organs, so young bud, mature petal, the flat-celled species have hardly any or no detectable expression of Mixture-like genes compared to the conical-celled species. And so we're seeing parallel evolution here, repeated loss of the same trait through repeated loss of expression of the same transcription factor in those tissues. Now, if these genes are expressed in leaves, certainly the Solanum dulcamara ones are, and these ones also seem to be, it's interesting to ask whether they have a role in leaves. And again, we can transform some of these species. So here's Solanum dulcamara made transgenic plants overexpressing its two Mixture-like genes, 1 and 2, or with them both knocked out through an RNAi construct. And what you're looking at here is leaf trichome frequency, so the number of leaf hairs that are produced. And if we overexpress either gene, we dramatically increase leaf hair number. If we take them out, then we almost lose leaf hairs completely. So in this system, at least, we're seeing a dual effect. We've lost the conical petal cell form through loss of expression of the gene in the petal, but we've gained leaf trichomes as a result of its expression in the leaf. Now, I said at the beginning that it was possible that this could be about selection, or it could be about genetic drift. And I just want to show you, not the data, but a movie that illustrates that we think, actually, this repeated loss of the trait is about genetic drift. So here's a bee foraging on another artificial replica flower, so we can make duplicates with conical or flat petal epidermal cells, but it's got a Solanum anther cone protruding through a gap in the middle. So it's a genuine Solanum reproductive set of organs here. If you look at what the bee's doing, there's hardly any contact with the petal at all. The contact is almost entirely with the anther cone. You occasionally get a foot slipping onto the petal, but the focus of the pollination system is entirely about the anthers. And so our preliminary data on this say that in fact the bees have no preference at all for conical or flat cell petal. It doesn't affect their foraging efficiency, it doesn't affect their behaviour in any way whatsoever. And so we assume in this system we're seeing loss of the trait through genetic drift, and then the interesting question for us is how often it's then been converted through into the production of leaf trichomes by a different promoter mutation in the Mixture gene. So I just want to finish by sort of run you through a fairly diverse range of stuff, but I wanted to finish by saying I hoped I've convinced you that you need to understand something about development and function if you're going to understand diversity and the evolution of that diversity. And to end by thanking people who've been involved in the work. I've shown you today primarily work done by Heather Whitney when she was a postdoc in the lab, by Katrina Alcorn, who's responsible for all the Solanum work, and by Edwige Moirud here, who's responsible for all the Hibiscus work. Lots of sponsors and collaborators, and thank you very much for listening.
Beverly, what are bees actually seeing? I mean, you were talking about visible light, but how much, say, of other ranges, infrared and polarized light, those conical cells, will they have an effect on the amount of polarized lights bouncing back off those particular surfaces?
Yeah, it's a good question. So we have to think quite hard about how the bees perceiving everything we work with. They're not seeing infrared, they're seeing ultraviolet, so their perception is shifted blue from ours. They don't see well into the red, or at all really into the infrared, but they see UV. Polarization is something that bees are traditionally thought to only see with the top part of the eye, and then it's involved in direction finding, looking at light coming off the sun, rather than direct interactions with flowers, although I think that's something that people are thinking about a bit more at the moment. The conical cells actually, and I didn't say this, work as very effective light scatterers. So iridescence is incompatible with having conical cells. I can only find iridescent species with the petal epidermis is smooth, because the conical cells themselves on that bigger scale scatter light so effectively that any optical effect, be it polarization or differential reflection of different wavelengths, is completely lost. So actually they're just a very good smoother out and evener out of the petal surface. And if you look at most flowers, they have that sort of velvety look that's because of those cells.
It looks like from your Solanum cladogram that what happens is once you lose it, the entire lineage no longer has conical cells. So have you seen any instances of gain of conical cells again?
Well, so there are 1,500 species or so, you tell me.
Just a few.
Just a few. We haven't got all of them character mapped on there yet. We're at about 15%, which is pretty good for the size of the genus, and it's focused in regions where the phylogeny is better known and where the species are more accessible. I think there are gains, but I'm not as confident in those gains as I am in some of the losses.
Mm-hmm.
It depends how well populated the phylogeny is with character states in order to give us confidence in that.
Very good. Thanks very much indeed.